In Chapter 9, we are going to discuss cellular communication. All right, so we are going to dive right into the different categories of cellular signaling. But before we start to talk about these different categories, there are some essential terms that you all need to be familiar with. So any type of communication that occurs between cells, so the communication that we are seeing in the signaling across the gap junctions, in paracrine signaling and endocrine signaling, that's communication between two cells. That is termed intercellular signaling, INTER, intercellular signaling. That's different than what we see in autocrine signaling. In autocrine signaling, this is known as intracellular signaling. And an easy way to remember this is that in Latin, these prefixes mean different things. Inter means between, or between two cells, and intra means inside, like intravenous injection, within the vein, right? Okay, so intercellular communication and intracellular communication. So endocrine, paracrine, and the gap junction signaling, that's all going to be dealing with intercellular communication, and the autocrine is going to be dealing with intracellular communication. So how do cells actually communicate? Well, cell signals are released by signaling cells, and you can see a lot of different signaling cell examples here: signaling cell, signaling cell, signaling cell. And these are the chemical signals that are released by these signaling cells are usually small, volatile, or soluble molecules called ligands. So you see all the ligands here. These are the ligands, these are the ligands, these are the ligands, okay? So a ligand is a molecule that binds another specific molecule, in some cases delivering a signal in the process. So you can think of ligands as the signaling molecules. And ligands are going to interact with their target cell, hopefully. So these are the cells that are affected by the chemical signals. And there are specific proteins that the ligands can bind to, known as receptors, and I will show you on the next slide in the presynaptic, postsynaptic neuronal communication example how the ligands bind to their particular receptors. And ligands and receptors, they exist in several varieties, but usually a specific ligand will have a specific receptor that typically only binds that ligand. It's kind of like that lock and key mechanism that I talked about in a previous chapter. All right, so now that we understand the differences between intercellular and intracellular, we know what ligands are, we know what signaling cells are, we know what receptors and target cells are. Now let's get into the different categories of signaling. So again, this slide is really important. I can ask a lot of test questions on this slide. So first, let's talk about autocrine signaling. So auto means self. So this is a form of intracellular communication, autocrine signaling. So autocrine signals are produced by signaling cells that can also bind to the ligand that is released. So this type of signaling often occurs during the early development of an organism to ensure that cells develop into the correct tissues and take on the proper function. So this is the type of signaling that we see in the cell death signaling pathways, or the apoptotic pathways. Apoptosis means programmed cell death. And why would that be important? Why would a cell want to kill itself? Well, let's say a cell is infected with a virus. Let's say there's this viral pathogen that comes into the cell. There can be autocrine signals that basically tell the cell to undergo self-destruction, so the virus inside the cell will die too. Remember, viruses are not living, and once they don't have that host, they are no longer capable of reproducing. So autocrine cell signaling is really important in the cell death pathways, or the apoptotic pathways. Another type of communication is when the cellular signal just jumps across a gap junction, or this communication bridge between two cells. And the ligand is going to jump across the bridge from the signaling cell to the target cell. And that is another type, so direct signaling across gap junctions. Intracellular mediators that allow small signaling molecules to move between cells. So Talked about autocrine, talked about the gap junction signaling. Now let's move on to paracrine signaling. So paracrine signaling is mediated by diffusion through the extracellular matrix. And signals that act locally between cells that are close together usually use the paracrine signaling mechanism. These types of signals usually elicit quick responses that last only a short period of time. So in order to keep the response localized, paracrine ligand molecules are normally quickly degraded by enzymes or removed by neighboring cells. Removing the signals will reestablish the concentration gradient for the signal, allowing them to quickly diffuse through the intracellular space if they are released again. So I'm going to go into a lot of detail on the next slide about the paracrine signaling mechanism that is used in neurotransmission. So I can ask you, just a general question. I can say, which of the following signaling mechanisms is predominantly seen in the apoptotic pathways? And I hope you tell me autocrine signaling. Or I can ask, which signaling mechanism is used by neurotransmitters and presynaptic and postsynaptic cells? I hope you tell me paracrine signaling. And then finally, we move down here to endocrine signaling. So endocrine signaling is going to be a lot slower than paracrine signaling, but the effects are going to last a lot longer. And the reason it's slower is because the signals in endocrine signaling, they have to travel to distant cells. And usually what happens is they have to go through the bloodstream. So this is how hormones are actually transmitted and how hormonal expression and hormonal regulation occur. And hormones, as you know, if you have a lot of testosterone, that's going to help you build muscle mass. If you have more estrogen, you are going to have more female characteristics. So with a slower response, because they have to go a longer distance, hormones do have a long-lasting effect. So again, the paracrine is going to be very close, very short-lived effect. Endocrine, there's going to be a long distance that needs to be traveled, but there is going to be a long-lasting effect. It wouldn't do us very good if all the testosterone or all the estrogen in our bodies was degraded very rapidly, or if growth hormone or insulin like growth factor 1 were degraded very rapidly. We would have a major imbalance in homeostasis. So those are the 4 categories of signaling. Make sure you know the differences between them and make sure you know examples of each of them. This is the paracrine signaling example that I was referring to on the previous slide for neurotransmission. But before we get into actually what's going on in this slide, again, there are some more terms that you need to be familiar with. So let's begin by talking about the presynaptic cell. They call this the presynaptic cell because this is the cell that does all the work before the synapse, before this gap between cell number 1 and cell number 2. So this is the presynaptic cell, this one up here. And this is going to be the postsynaptic cell, cell number 2. And then this is the synapse, the gap between the two. Okay? Presynaptic, postsynaptic, and synapse. And then we have neurotransmitter. There are a whole bunch of different types of neurotransmitters, and the one I'm going to talk about on this slide is known as acetylcholine, or ACH. So acetylcholine is really important for muscle contraction and muscle integrity. If we don't have acetylcholine at the neuromuscular junction, well, then muscles aren't going to work properly. So let's imagine all of these little neurotransmitters in these vesicles are acetylcholine neurotransmitters. So in the presynaptic cell, what's going to happen is the neurotransmitter is going to be bundled and bunched up into a vesicle. The vesicle is going to move to the presynaptic membrane, and then once it reaches the presynaptic membrane, exocytosis is going to occur, and the neurotransmitter is then going to be released into the synaptic cleft. Okay? 3 really important things can happen at the synaptic cleft. Number 1, it's written right here for you. The neurotransmitter can attach to the receptor that it wants to. So this is the ligand binding to the receptor of interest. And the signal can propagate down and continue about its merry way. The second thing that could happen is written right here. There could be particular enzymes that destroy the neurotransmitter. And these enzymes aren't always bad, because if we have too much neurotransmitter, or if we have too much neurotransmitter, then that could disrupt homeostatic balance. So enzymatic destruction can be helpful. Enzymatic destruction can also be inhibitory if the enzymes become overactive and start to destroy the amount, leaving us with the desired amount, leaving us with less than desirable amounts of neurotransmitter. And that's kind of what happens in some autoimmune diseases like myasthenia gravis. One of the theories behind myasthenia gravis is that the acetylcholine neurotransmitter at the neuromuscular junction gets degraded too fast, and it gets degraded by this enzyme known as acetylcholine esterase. Esterases break down proteins and molecules. So in that case, it's bad for too much enzymatic destruction. And finally, the third thing that could happen is the neurotransmitter can be reuptaken back into the presynaptic cell and then degraded by what are known as MAOs, or monoamine oxidases. And this begins to get us thinking about some of the ideology behind depression. One of the main neurotransmitters that's thought to be disrupted in depression is serotonin. And one of the theories is that people who are experiencing depression have lower serotonin levels than people who are not experiencing depression. So one of the most famous antidepressant drugs is Prozac. Prozac is known as an SSRI, or a selective serotonin reuptake inhibitor. So what Prozac actually does is, once the neurotransmitter, we're going to change from acetylcholine to make this serotonin now because we're talking about depression. Once the neurotransmitter serotonin gets released into the synaptic cleft, the SSRI, selective serotonin reuptake inhibitor, it's going to block the reuptake. It's going to block this presynaptic pump, allowing more neurotransmitter to remain in the cleft. Therefore, ideally, more neurotransmitter can bind to the receptor and more proper cellular signal propagation can occur. Selective serotonin reuptake inhibitor, we're blocking that reuptake pump. Okay? So make sure that you're familiar with the terms. Make sure that you're familiar with the fact that neurotransmission is one of the best examples of paracrine signaling, and understand the 3 events that could happen at the neuronal synapse. Internal receptors can also be called intracellular or cytoplasmic receptors, because that's where we find them. We find them in the cytoplasm. So again, just kind of orienting you all with this slide. Out here is the extracellular fluid. Here's our signaling molecule, or our ligand. And then inside the cell, once the ligand enters through the plasma membrane, it then goes into the cytoplasm, where this ligand receptor complex can form. So this is the ligand receptor complex. So what is this important for? Well, this is really important for DNA synthesis. If we don't have DNA synthesis, we never make protein, and we never have the potential to maintain homeostatic balance in our bodies. And that would be very, very bad. So once inside the cell, this one here, we're talking about hydrophobic signaling molecules that are going across the plasma membrane. That's going to be our ligand. So we're talking about hydrophobic signaling molecules as our ligand. They're going to diffuse across the membrane, and they're going to interact with these intracellular receptors in the cytoplasm. And once that happens, once we have this ligand receptor complex, this can help us mediate gene expression. Gene expression is the cellular process of transforming the information in a cell's DNA into a sequence of amino acids, which ultimately forms a protein. So when we have this complex again, what that complex actually does is it creates a series of events which lead to a conformational change. And that conformational change allows for not only migration into the nucleus, but also the freeing up or the opening of a DNA binding site where this ligand receptor complex can bind to. So it binds to this specific regulatory region on chromosomal DNA, and it actually promotes the initiation of transcription in this example. And remember, the central dogma, we've talked about that previously. It's really important to understand that DNA is transcribed into RNA, and RNA is translated into protein. So without these internal receptors, there are no way for these hydrophobic signaling molecules to enter into the cell and for them to form these ligand receptor complexes and integrate into our DNA. Cell surface receptors can also be called transmembrane receptors, and as the name implies, they are found on the cell surface, and they are membrane-anchored proteins that bind to external ligand molecules. And we can find this type of receptor spanning the entire plasma membrane, and it's responsible for performing signal transduction. So ligands that interact with the cell surface receptors do not have to enter the cell that they actually affect. Cell surface receptors are also called cell-specific proteins or markers because they are specific to individual cell types. So now let's talk about this picture, what's actually going on in this picture. So what we're looking at here is a gated ion channel. And these channels are found in the plasma membrane, and they allow for the controlled flow of ions into and out of the cell. So let's look at what's going on in figure A here, okay? So I'm just outlining the channel. The channel is closed. So we have our ligand in orange, and then we have a bunch of ions in yellow, okay? So there's no way right now for any of these ions in yellow to go from the extracellular fluid into the cell because the gate is closed. But what happens is that a signaling molecule, the ligand, is going to come and bind to the cellular surface, and it binds to the channel protein, and then what happens is a conformational change occurs and that allows for the gate to be open. When the gate is open, the ions can rush inside the cell. And then in C, what we see here is we take this signaling molecule and we remove it from the binding site. And when the signaling molecule is released and removed, again, a conformational change happens that closes this gate. But we still see this ion concentration in here, but this conformational change allows us to balance the amount of ions inside and out of the cell. So again, these are very complex processes that are happening constantly throughout our body. And thank God we don't have to think about them consciously, or we would be very consumed in very intricate thought processes all the time. We have intracellular and intercellular mechanisms and signaling pathways that moderate all of this for us. G protein linked receptors bind a ligand and they activate a membrane protein called a G protein. The activated G protein then interacts with either an ion channel or an enzyme in the membrane, and that's what we're going to talk about in this picture here. All G protein linked receptors have 7 transmembrane domains, but each receptor has its own specific extracellular domain and G protein binding sites. Cell signaling using G protein linked receptors occurs as a cyclic series of events, and that's what we see in the picture here. Before the ligand binds, and that's what this picture up here is going to be talking about, the inactive G protein can do a few things. It can bind to a newly revealed site on the receptor specific for its binding, or it can just remain inactive. Once the G protein binds, if it does, to the receptor, the resulting change in shape activates the G protein. Once the G protein is activated, the guanosine diphosphate, or GDP, is going to be released, and the complex is going to pick up GTP, or guanosine 3-phosphate, GTP. The subunits of the G protein then are going to split into the alpha subunit and then the beta gamma subunit. One or both of these G protein fragments may be able to activate other proteins as a result of this splitting. So that's what we see here. A signal molecule comes in and it binds to the receptor, and there is the exchange of GTP and GDP. And here is the dissociation. The dissociation of the alpha complex and the beta gamma complex. The alpha complex is going to have that GTP, and when that occurs, conformational change occurs, and that elicits a cellular response. And then what's going to happen is GTP can be hydrolyzed to GDP, so that's just basically losing a phosphate group, so we go from GTP to GDP, and then that unit is going to dissociate from the receptor, and then a new cycle begins. There's going to be an open site on top of the receptor, and there is the potential for another signaling molecule to come in and bind and do the whole GDP-GTP exchange and the dissociation of the subunits again, and sequentially the triggering of the cellular response. On the next slide, we're going to talk about enzyme-linked receptors, but before we talk about them, there are some more terms that you need to know and understand. So, signal transduction. When a ligand binds to a receptor and the signal is transmitted through the cell membrane and into the cytoplasm, continuing the signal. So we saw an example of signal transduction on the previous slides, where we have that ligand receptor complex that is formed in the cytoplasm. Remember, the ligand starts in the extracellular component, and then it can cross into the plasma, into the cell via crossing the plasma membrane, and then it makes that ligand receptor complex, which eventually can move into the nucleus when conformational changes occur. This next term here, dimerization, this is going to be really important to understand what is actually going on in the receptor tyrosine kinase enzyme-linked receptor on the next slide. So dimerization, two receptors bind to each other to form a stable complex. So addition. And then there are a lot of different signaling pathways. These are chain of events, including second messengers that we talked about, the G-coupled protein. G-coupled proteins are second messengers. We're going to talk about the enzymes on the next slide. The receptor tyrosine kinase is an enzyme-linked receptor, which you're going to learn all about, and activated proteins that follow ligand binding to a receptor. And then we have signal integration. So signals from two or more different cell surface receptors merge to activate the same response in the cell. So two signals integrate, they combine, and they do something in a similar fashion. They merge to activate the same response in the cell. They can accelerate pathways together, they can decelerate pathways together, but the important thing is they work together. Now let's talk about enzyme-linked receptors. Enzyme-linked receptors are cell surface receptors with intracellular domains that are associated with an enzyme. Sometimes the intracellular domain of the receptor itself can be an enzyme. Other enzyme-linked receptors have a small intracellular domain that interacts directly with an enzyme. And the enzyme-linked receptors normally have large extracellular and intracellular domains, but the membrane-spanning region consists of a single alpha helical region of the peptide string. Now let's talk about what's actually going on in this picture here so we can understand all of this scientific jargon in a visual sense. All right, so what we're doing is we are looking at a receptor tyrosine kinase. And this is an enzyme-linked receptor with a single helical transmembrane region and extracellular and intracellular domains. So we have the alpha helix, and there are extracellular components, the extracellular domain, and there are intracellular components, part of the intracellular domain. So what actually happens here is this signaling molecule binds to this receptor tyrosine kinase. It binds, again, this is that lock and key binding. And once it binds, it causes this receptor to undergo what is known as dimerization. So dimerization, or the bonding and binding of these two receptors. So here in figure 1, you can see that they're split. Once the signal molecule comes on, it causes dimerization, and these two will fuse together. In figure 3 down here, the tyrosine residues on the intracellular domain are then autophosphorylated. So again, how do we phosphorylate things? We use ATP. ATP is going to donate the phosphate to the different tyrosines, and because we're losing the phosphate off ATP, we get ADP. And this happens 6 times. So the picture right here, this is a fully activated receptor tyrosine kinase. All of the tyrosines are completely phosphorylated because of the 6 processes of ATP phosphorylation. And then in figure 4, what's going to happen is the triggering of cellular responses. All types of different cellular responses can be triggered. And then the signal eventually is going to be terminated by a phosphatase that is going to remove the phosphates from the phosphotyrosine residues. So these phosphatases are depicted in purple here. They're going to relay here, and they're going to remove all of the phosphate here to render this inactive. Over time, as it becomes more and more inactive, the dimerization is going to dissociate. The signaling molecules are no longer going to be attached, and this whole process has the capability to start over. Ligands can act as chemical signals that travel to target cells to coordinate certain responses. The types of molecules that serve as ligands are incredibly varied, and they range from small proteins to small ions like calcium. Here we're going to talk about small hydrophobic ligands. And these have the capability to directly diffuse through the plasma membrane and interact with internal receptors. Important members of this class include the steroid hormones. So you can see the chemical structure of the steroid hormones, estradiol and testosterone. They have the exact same carbon cyclic backbone as cholesterol does. So cholesterol is a precursor to the steroid hormones, estradiol and testosterone. So steroids are lipids that have a hydrocarbon skeleton with 4 fused rings. And different steroids are going to have different functional groups attached to that carbon skeleton. So you can see in estradiol, there's a hydroxyl group coming off here, and there's just a double-bound oxygen coming here. And there are different methyl groups in testosterone. Again, we see a methyl group branching here, and that is going to be absent in estradiol. So depending on the rest of the steric configuration, that is going to give the particular hormone its particular function. So there are many other hydrophobic hormones. I just mentioned some of the steroid hormones, but there's other ones that include thyroid hormones and vitamin D. And it's important to note that in order to be soluble in blood, hydrophobic ligands must bind to carrier proteins while they are being transported through the bloodstream. On the definition slide, I talked about signaling pathways, and I wanted to give you an example of a signaling pathway. So on this slide, don't worry about memorizing everything here. Scientists, they dedicate their entire scientific careers to studying this epidermal growth factor pathway. So again, you don't need to memorize things here. This is just an example. The epidermal growth factor receptor, or EGFR, is a receptor tyrosine kinase involved in the regulation of cell growth, wound healing, and tissue repair. So what's generally happening in this pathway is that EGF, or epidermal growth factor, binds to a particular receptor, and this whole cascade of downstream events occurs, and that's what's responsible for allowing the cell to grow and divide. So I can ask you a test question on the role of the epidermal growth factor and what does this pathway actually do. So the answer is right here. Helps regulate cell growth, wound healing, and tissue repair. If EGFR is activated at inappropriate times, uncontrolled cell growth may occur. So again, scientists dedicate their entire lives to understanding this because there is a lot of direct linkage to oncology and cancer. Cells render themselves anti-apoptotic. They don't die when they're supposed to. They grow out of control. And scientists believe that this is due to improper EGFR signaling. This is a really cool video on cell signaling, so please take a moment, pause the PowerPoint at this point, and watch this video. So we've spoken briefly about second messengers in the form of the G-coupled protein receptor example that I gave you, but this is a very important concept and I would like to reiterate it again. So second messengers are small molecules that propagate a signal after it has been initiated by the binding of the signaling molecule to the receptor. Okay, so we've talked about the ligand binding to the receptor and then conformational changes happening and then the downstream signaling events happen. That's where second messengers come in. They help to facilitate the signal after that ligand receptor complex has occurred. And this diagram shows the mechanism for the formation of CAMP, or cyclic AMP. So I hope you all recognize this molecule here on this side in the form of the adenine, the ribose, and the 3 phosphates, adenosine triphosphate, or ATP. And then through this enzymatic reaction, ATP can become CAMP. And CAMP is really important to both activate and inactivate proteins within our cell. And the termination of the signal occurs when an enzyme called phosphodiesterase converts CAMP into AMP, or adenosine monophosphate. So remember this esterase here. Where did we see that before? Well, we saw that when we talked about the neuronal communication and the synapse, where the acetylcholine esterase would be present to break down acetylcholine. So phosphodiesterase, what that's going to do is that's going to be removing some of the phosphate groups and allowing for the conversion of CAMP into adenosine monophosphate. Present in small concentrations in the plasma membrane, the inositol phospholipids are lipids that can also be converted into second messengers. Because these molecules are membrane components, they are located near the membrane-bound receptors and can easily interact with them. Phosphatidylinositol, or PI, is the main phospholipid that plays a role in cellular signaling. Enzymes known as kinases phosphorylate PI, or pi, to form pi phosphate, PIP, and pi bisphosphate, PIP2. The enzyme phospholipase C has the capability to cleave PIP2 to form diacylglycerol, or DAG, and inositol triphosphate, or IP3. These products of the cleavage of PIP2 serve as second messengers. DAG remains in the plasma membrane and can activate protein kinase C, which then can phosphorylate serine and threonine residues in its target proteins. And the inositol triphosphate, or IP3, has the capability to diffuse into the cytoplasm and bind to ligand-gated calcium channels in the endoplasmic reticulum to release calcium to continue signal cascades. Some signal transduction pathways regulate the transcription of RNA. Others regulate the translation of proteins from mRNA. An example of a protein that regulates translation in the nucleus is the MAP kinase ERK, or ERK. The MAPK-ERK pathway, also known as the RAS-RAF-MEK-ERK pathway, is a chain of proteins in the cell that communicates a signal from a receptor on the surface of the cell to the nuclear DNA. ERK is activated in a phosphorylation cascade when EGF, or epidermal growth factor, binds the EGFR or the EGF receptor. And upon phosphorylation, ERK can enter into the nucleus and activate a protein kinase that in turn regulates protein translation. The result of signaling pathways can affect muscle cells. The activation of the beta-adrenergic receptors in muscle cells by adrenaline can lead to an increase in cyclic AMP inside the cell. Adrenaline is also known as epinephrine. It's a hormone that's produced by the adrenal gland, which is found on top of our kidneys. And what that does is it helps our body prepare for short-term emergencies. So I'm sure you've heard the term fight or flight, the activation of the sympathetic nervous system. Well, what is allowing us to either fight or run very fast away from a situation is the surge of adrenaline that's coming from the adrenal glands. Cyclic AMP has the potential to activate protein kinase A, or pKa, which in turn phosphorylates two enzymes. The first enzyme promotes the degradation of glycogen by activating the intermediate glycogen phosphorylase kinase, that in turn activates glycogen phosphorylase, which catabolizes glycogen into its constituent glucose. And we've talked about that in the form of glycolysis. And then we have cell growth. Cell signaling pathways also play a major role in cell division. Cells do not normally divide unless they are stimulated by signals from other cells. The ligands that promote cell growth are called growth factors. Most growth factors bind to cell surface receptors that are linked to tyrosine kinases. These cell surface receptors are called receptor tyrosine kinases, or RTKs. The activation of RTKs initiates a signaling pathway that includes a G protein called RAS, or RAS, which activates the MAPK pathway that we talked about earlier. The enzyme MAPK then has the potential to stimulate the expression of proteins that can then interact with other cellular components to initiate cell division. So again, you don't have to memorize everything on this slide either. These are just some responses to cell signaling, and I hope you are feeling a little overwhelmed because this is a lot of information, and I want this information to resonate with you, and I want the complexity of this information to resonate with you because biology is a very complex subject, and there are a lot of signaling events that happen that we don't even realize and we just take for granted. But what these slides are doing is they're highlighting the importance of these signaling events. What happens if adrenaline wasn't activated in a fight or flight response? Well, he might be dead. What happens if there is no capability for our cells to divide? Well, we have to have cell division in our body to maintain growth and homeostasis. So again, we would die. Very important mechanisms. I like this slide a lot because it just fascinates me how yeast cells communicate and the purpose of their communication. So yeast, they are eukaryotic organisms. They're actually fungi. And the components and processes found in yeast signals are very similar to those of cell surface receptors that we find in multicellular organisms. So they may have some type of intelligence. Who knows? Budding yeast, which we see in the picture here, are able to participate in a process that is similar to sexual reproduction that entails two haploid cells combining to form a diploid cell. In order to find another haploid yeast cell that is prepared to mate, these budding yeasts secrete a signaling molecule known as mating factor. When mating factor binds to cell surface receptors and other yeast cells that are close by, they stop their normal growth cycles and initiate a cell signaling cascade that includes protein kinases and GTP binding proteins that are similar to G proteins. So they are communicating in order to mate and further bud. Signaling in bacteria enables bacteria to monitor extracellular conditions to ensure that there are sufficient amounts of nutrients and ensure that hazardous situations are avoided. There are circumstances when bacteria can communicate with each other. The first evidence of bacterial communication was observed in a bacterium that has a symbiotic relationship with a particular squid out in Hawaii called the Hawaiian bobtail squid. When the population density of bacteria reaches a certain level, specific gene expression is initiated, and the bacteria produce bioluminescent proteins that have the capability to emit light. Because the number of cells present in the environment, cell density, is the determining factor for signaling, bacteria signaling was named quorum sensing. In politics and business, a quorum is the minimum number of members required to be present to vote on an issue. Quorum sensing uses autoinducers as signaling molecules. Autoinducers are signaling molecules that are secreted by bacteria to communicate with other bacteria of the same kind. The secreted autoinducers can be small, hydrophobic molecules such as acyl homocystine lactone or larger peptide-based molecules. Each type of molecule has a different mode of action. Some species of bacteria that use quorum sensing form biofilms. Biofilms are responsible for the contamination and the proliferation of bacteria on prosthetic devices. Bacterial biofilms can invade implants, and they can invade heart pacemakers and can cause life-threatening infections.